Hi, my name is Gil Zaltzman and I am ECNP Chair of Education. I am an adult and child psychiatrist. The organ we study and treat as psychiatrists is the brain. We know today that the whole phenomena of thinking, feeling and behaving arises as a consequence of electric and chemical activation of neurons and networks across the brain. Let's start in the beginning. During pregnancy, the fetal brain is developed from a cell layer called ectoderm that gives rise to epithelium and the nerve system. During the third week of development, the ectoderm becomes thickened and forms the neural plate and the neural groove in its middle. The groove is creating neural folds and crest. These structures will develop on day 28 to give the neural tube. Here you can see the structures and the cells that will be created at that stage. This tube will later create the three main structures: forebrain, midbrain, and hindbrain. The forebrain is the source of the two hemispheres, the basal ganglia, hippocampus, and thalamus, while the hindbrain will create the cerebellum. According to evolution theory, the human brain is similar to most mammalians. However, there are some differences, like the size of the prefrontal cortex, that account for a third of our brain volume and only 5% of the dog brain and 10% of the monkey brain. Human brain at large may be seen as three layers of development and complexity. The primitive brain, that we share with most creatures, is located here and is responsible for self-preservation activities such as eating, drinking, temperature regulation, aggression, blood pressure, and breathing. The intermediate brain that is responsible for emotion regulation and consists of the limbic system that was discovered in the 30s. The neocortex, the rational brain that is responsible for higher intellectual functions such as executive functions. This cortex is highly developed in primates. Let's go into microanatomy of the brain. The human brain contains approximately 85 billion neurons. Neurons are composed of cell body with the nucleus, the metabolic factory of the cell, the dendrites, little antennas that are branched out and get input from other neurons, the axon that moves information to other neurons, and the axon terminals, including the synapse, that release neurotransmitters as messengers to other neurons. These transmitters influence brain circuits and our behaviors and will be discussed later. Other cells that can be found in the brain are glial cells, such as oligodendrocytes, Schwann cells, microglia, and astrocytes. Astrocytes are especially interesting, and their influence and role in psychopathology was previously underestimated. We know today that astrocytes have an important role in synaptic transmission and special role in the functioning of some structures like the hippocampus. The microglia were thought in the past as having a role in neural death, but some new data show a regulatory function and part in neurogenesis. In postmortem studies in patients with schizophrenia, decreased number of glial cells was demonstrated in the dorsolateral prefrontal cortex and in depressed patients in the medial prefrontal cortex. At large, cell bodies are located in the gray matter, axons travel into the white matter. The brain is organized as circuits and networks. This is an example of three circuits. Let's move from microanatomy to the brain at large. This is our scalp, in which the brain is located. This is a model of a normal human brain. This is the actual real size of adult brain, and its weight is about 1.5 kilo. We have right and left hemispheres, and we have four lobes: the frontal lobe, the parietal lobe, the temporal lobe, and the occipital lobe. The cerebral cortex of each hemisphere is divided into 4 major regions, again the frontal, parietal, temporal, and occipital. Here is an animation of the different lobes. Cytoarchitectonic subdivisions of the human brain, as determined by Brodmann, are still used today. The prefrontal cortex, Brodmann 9, 10, 11, and 12, is the youngest and the most developed part of the human brain and the source of our intellectual activity. It can be divided into dorsolateral and ventrolateral regions, with each of these regions having different functions. The dorsolateral prefrontal cortex, that is the location of some major psychopathologies, seem to be more involved in the manipulation of data during working memory tasks and other high executive functions. The Broca motor language area is located at the inferior part of the frontal lobe, while the Wernicke sensory part, responsible for comprehension of language, is located at the temporal cortex. Interestingly, in babies, this area is developed before the Broca, which means babies understand language before they can talk. Beneath the cortex are the basal ganglia structures, the caudate nucleus, the putamen, and the globus pallidus, important in controlling movement and posture. These nuclei are interconnected. The substantia nigra is the source of dopamine. Our D2 blockers may influence these areas and create severe motor side effects. The limbic system is connected to our emotional life and consists of the cingulate gyrus, the hippocampus, the hypothalamus, and the amygdala. The amygdala controls mood, fear, rage, and aggression. The cerebral hemispheres contain billions of myelinated axons or fibers. These axons are bundled into white matter tract, shown here in a DTI colorful imaging. MRI and DTI studies have shown abnormalities in white matters in schizophrenia, depression, Alzheimer's disease, and in children with autism. The three-dimensional structure of the brain may be confusing, so let's try to look on the relationship between the parts using a model of the human brain and some animations. Midbrain, pons and medulla, cerebellum, thalamus, hypothalamus, hippocampus, ventricles, caudate nucleus, putamen, amygdala, and finally, the white matter and the gray matter. In the past decade, Jay Gide and others have shown using MRI on normal developing kids that the brain maturation process is not done before age 22. This normal process is called pruning. Gray matter is growing in volume until puberty and then going through a cut down of unmatured gray matter as a normal process. This short clip shows that pruning starts in the occipital part and gets to the frontal part only at the end of adolescence. This may explain why teenagers may lack some inhibitions and reasoning. Interestingly, this process is severely disturbed in adolescents with major psychiatric disorders. I hope we are now on the same page in basic functional neuroanatomy and can move forward to the next learning unit. Thank you.